So good afternoon and welcome back. We're ready to commence the next session, which is our roundtable discussion on benefit-risk of cancer immunotherapies. We felt that this is a topic that has not been discussed yet, since the immunotherapy field is basically entering into its renaissance. And now that we have two new modalities that are quite distinctly different types of immunotherapies which are approved, which are being used in a commercial setting, and for which there is a lot of clinical data, either from clinical trials or in the commercial setting. It is probably the right time to initiate a discussion about benefit-risk, which might actually turn out to be a bit different than what we're used to with standard therapies, namely chemotherapies or targeted agents. And then there's a second component to this, which obviously would have to be how the integration of the monotherapy approach for which these two new treatments were approved, how that will integrate into the standard of care, meaning how it combines with other modalities. And certainly that may impact on the perception of benefit risk. And the treatment community or the treater community would certainly benefit from getting a flavor how perception works with cancer immunotherapies now. It will impact on how probably development might be done going forward. So having said all this, we have composed a round table, or actually in this case a panel, I would say, of experts that all have experience with treating patients and developing cancer immunotherapies, and can certainly provide a perspective on the concept of benefit risk. So with that, I hand it on to Jill O'Donnell, and then we do introductions.
Well, thank you, Axel. Again, I'd like to thank all the panelists. As Axel has said, we have brought together both some very notable medical oncologists and some notable cancer immunologists. So hopefully we're having differing viewpoints here to really discuss the benefit and risks that immunotherapies bring to what we hope will eventually become the real standard of care for cancer. And we will use these the discussion points that come out of today's panel to really help guide both the Cancer Research Institute and the Cancer Immunotherapy Consortium to decide on future things we should be discussing, actions we should take, or practical things that we could put into play to help answer or advise on how we handle these risk benefits and how to make it as beneficial as possible for patients. So with that, I guess I'll introduce myself and then we'll go down and introduce, for those of you that don't know me, Jill O'Donnell Tormey, CEO and Director of Scientific Affairs of the Cancer Research Institute.
I'm Axel Huus. I'm the Vice President for Research and Development for Combination Therapies at GlaxoSmithKline and also Co-Director of the Cancer Immunotherapy Consortium.
James Gully. I'm a medical oncologist at the National Cancer Institute, do prostate cancer immunotherapy clinical trials.
Phil Kantoff. I'm a medical oncologist at the Dana-Farber Cancer Institute.
Vincent Brichard, responsible for therapeutic vaccines at GSK.
Jedd Wolchok, Memorial Sloan Kettering Medical Oncology, and I'm also the director of the CVC.
Susan Slovin, genitourinary oncologist at Memorial Sloan Kettering.
Jim Allison, I'm the immunologist at Memorial Sloan Kettering. I'm head of the immunology program and director of the Ludwig Center there. And I do basic stuff mostly.
Yeah. I'm Larry Norton, Memorial Sloan Kettering, and I work with the rest of these guys.
All right, so if I may say that, you might have noted there is a strong presence of Memorial Sloan Kettering here at the table, which is actually more or less accidental. We have chosen the experts based on their background that they have, and it so turned out that many of them come from Memorial. So in any case, I would like to begin the question and answer session, and I'd like to encourage the audience actually to also feel that you can ask questions. This is an open discussion. We will kick this off among the panelists, but there's no reason for the audience to feel passive. So you are more than welcome to throw in additional questions as it relates to benefit risk. So I will ask the first question, which is actually A general question for all the panelists where I would like to hear an opinion from each panelist, and we'll go around, about the perception of benefit risk. In the introduction we said we have 2 new immunotherapies now, distinctly different agents, an immune modulatory monoclonal antibody, ipilimumab, and a autologous dendritic cell vaccine, Sipaloy-Cel-T, that have distinctly different profiles. They're now available for some time, at least a year in the commercial space or more, and of course a lot of clinical trial data. So it'd be interesting to understand, how do you perceive the clinical profile of these agents, the use in patients, and the benefit risk altogether, and how does that reflect on immunotherapies in general? So why don't we begin on the left side here. Larry, would you like to take the first crack at this?
Yeah, well, I'll give you sort of a general view of the, from the medical oncology point of view. And by the way, when I gave my talk, I forgot to mention that Heather MacArthur is actually chairing the study that I mentioned. It's strongly influenced by Susan, who did the radiotherapy as a antigen release, and Jedd is extremely involved he's our IP expert really, and very involved in the design of the trial, as well. So it's a very team approach. I think right now, I think the community of patients is very excited of the prospects of immunotherapy, because it has a very kind of sexy aura about it, but also because of the success of immunotherapy in terms of prevention of infectious diseases. I think the concept is that you will have like an organ rejection kind of reaction where any disease in your body would react rather than what we see with infectious diseases. Its role is not therapy, but rather prevention. But that perception is obviously going to change. It's very early on, or it's going to morph in some way because it's very early on in the field. I think the main thing that we have to think about is that whatever we develop in patients with advanced disease is going to get pushed very, very quickly into patients earlier on, the adjuvant setting, the primary disease setting, and maybe even in the prevention setting if the experience in patients with disease, with overt disease is successful. So we're going to have to pay attention to long-term toxicities, which is a long-term consequence of manipulation of the immune system, which is, at the present time, I think, in my view, impossible to predict. There's no manipulation of biology that doesn't have some kind of long-term consequence. And what that is going to be and how that's going to appear is something we have to be very sensitive to. And the reason I'm mentioning this is because in the clinical trial community, there's a lot of pressure to get very quick results and then close your study for further follow-up. Because it's expensive. Long-term follow-up is very expensive. The cooperative groups, which are just a shadow of their former self, are moving toward very short-term follow-up. So response rates, response duration, the data that you saw with the Provenge vaccine, is that, it may not be the first response that may be important. It may be subsequent responses, and that's why patients may live longer. Phil's probably has addressed that and may make comments about that. So, there could be a long-term effect, and the long-term effect could be beneficial in patients with advanced disease, but it could be detrimental in patients without. So that part of it, we have to be very careful. And from the medical oncology point of view, we have to be extremely cautious about overhyping the field so that expectations are so high that unless we get really spectacular results, people are going to be disappointed relative to the expectations, and it's going to hurt the development of the field. That's the acute issue that we have to wrestle with. And the other issue we have to wrestle with is the one I just mentioned, long-term effects. We have to make sure that patients on clinical trials are followed sufficiently, in sufficient duration, sensitively enough that we can pick these things up and act appropriately and modify our trials appropriately. Because if we don't do that, we may end up with some very severe problems.
All right, Jim.
Okay, thanks. I've got obviously a different perspective coming from the basic science world. But what I can tell you is that with F.A.C.P, we've given F.A.C.P, we've treated thousands of mice over the last 15 years, including some of the guys who are here in the room in my lab, and we've never seen adverse events with the exception of melanoma, where we could see depigmentation, which is something that I guess you could expect. But in the early days, we treated mice chronically for months with very high doses of F.A.C.P, and other than occasionally picking up some low-level titers of anti-DNA antibodies and things like that. We never saw any evidence of mice getting sick. And this was also the experience of the early clinical development with all the toxicity studies that were done in cynomolgus monkeys at very high doses, very long, repetitive schedules of treatment. There was no toxicity in monkeys either, so it was a bit of a surprise when colitis and some other things began to appear in the patients. But I think that was scary early on when it wasn't expected because of the animal studies that preceded the clinical trials. And I think it initially caused a lot of concern. But for almost the real fear that it would cause autoimmunity or something just generally has not proven to be the case. And as you saw from Jedd's talk today, there are some toxicities, but they're sporadic and they're of different kinds. They're not necessarily predictable, but they're certainly not like the mechanism-based toxicities that you see with the old chemotherapies, which made everybody that got the drug very ill, and with predictable problems with that everybody knows more about than I do. So I think it's interesting with time, and I think Jedd could talk about this, with time and experience. Again, algorithms were developed for dealing with these, and I think that it's really become a lot less frequent than the adverse events were in the early days. So again, a surprise after the benign effects in the mice, but it seems to me that given the long-term benefit, 4.5 years in a fraction of the patients in melanoma, with at least one patient from the phase 1 trial that was 12 years ago is still alive without retreatment or without disease. I think the durable responses seems to me may be worth it.
All right, Susan.
Well, I think we all have tremendously renewed enthusiasm toward immunotherapy, and it's very gratifying to see patients come into clinic and want to know how to boost their immune system. So when you talk about patient perception, there really is a very somewhat skewed perception, because their idea is that, well, perhaps correctly in one way, they do have an altered immune system that makes them perhaps more susceptible to problems with immune surveillance. But on the other hand, they think that doing things like coffee enemas or eating 15 bananas a day or doing something that will change their immune system, or they have all these things out there. Thymus extract, that's the most popular one. Just put some in your milkshake in the morning or your soy milk, and you're going to regenerate your thymus and regenerate all your little T-cell precursors. That being said, the perception by the community is that it's not particularly toxic, it's very safe, which is appropriate. However, in the scientific community, I think we've all seen that using immunotherapy, particularly certain agents that we've alluded to, may not be optimally used as single agents. And we're starting to look at combo developments, which is, I think, increasing the responses that we're seeing with a lot of these newer agents. And that being said, we may be subjecting ourselves to seeing a lot more but tolerable side effects. I don't know that any of us were prepared to see some of the problems with colitis, although it is manageable. Jedd led the way in melanoma in terms of regulating the issues that we had in dealing with hypophysitis, colitis, transaminitis. But I think one thing has to be brought in to understanding, and again, we still are very young in our experience with these drugs, and that is, it may very well be that it's a very disease-specific type of toxicity, that there may be some diseases that lend themselves to have a little bit more sensitivity to the drug, with perhaps a little bit of a higher frequency of these side effects, albeit very manageable. And of course, if we could very early on identify those patients who may just be a little bit more susceptible, that'll be extremely helpful for us in going forward.
I don't want to be repetitious because a lot of important information was just communicated, but I think it's worth stating again that the public perception is that immunotherapy is something that's logical and that it shouldn't be toxic. I think that we know that the logic is there, but the lack of toxicity is something that we've learned is really different from reality, that people do have side effects from these manipulations, and the key is learning how to manage them in a simple and consistent way. because there is a trade-off, and the trade-off for any side effects is that there are people who benefit in a very durable fashion from these interventions. The long-term survival as the endpoint here is really a testament to that. And many of you may have seen, there was an article in the New Yorker this week about immunotherapy, which used some colorful language to describe some of the side effects of ipilimumab. I think the term atrocious diarrhea, which kills people, was thrown around. And I think we have to put that in the context of what we actually see on a daily basis, which is that, yes, people do get side effects, but we know how to manage them. And most people, we can manage this on an outpatient basis with very simple algorithms using commonly available medicines like prednisone. And Jim alluded to this, that in kind of summarizing our experience at Memorial, where we've probably treated about 300 patients with ipilimumab, we looked at the number of people that we had to admit to the hospital for management of side effects. And in looking at that over the period of time between, say, 2006 and 2009, within the first year, the number of people that we had to admit dropped by 50%. Because of familiarity with algorithms, development of algorithms, and I have to actually credit Axel and the team at Bristol at the time for helping us to develop said algorithms, because once we learned what these side effects were, I mean, as Jim said, the animal models were not predictive of these toxicities, because I think one simple fact is that these animals live in a much cleaner environment than we do. And so the influence of their microbiome and what they're exposed to in their environment is very different. We're much dirtier creatures, believe it or not. And so the toxicities that were seen in humans were different than what was seen in any of the animal models. So my message is that there is a benefit, but that practitioners, if they're alert and attentive and follow the guidelines, the trade-off is worth it.
Yeah, so I would just want to interject one piece. We have, of course, different types of immunotherapies. So far, I've heard a lot about ipilimumab, which has a distinct toxicity profile. There's a benefit to the drug as well. Now, if we look at Cipraloy-SLT, on the other hand, this toxicity profile is quite different. So maybe we can direct the next few comments on the other side of the spectrum.
So yeah, so I think when we come to looking at the risk-benefit profile, I think it's important to understand that there are differences between the risk-benefit profile for a therapeutic vaccine such as Cipraloy-SLT and what else we have in our armamentarium as medical oncologists, usually chemotherapy. So first of all, when we're defining benefit, I think what we've seen with these immune therapies, be it Cipraloy-SLT or ipilimumab, we see no improvement in median time to progression, but we do eventually see improvement in overall survival, which I would say is the best evidence of whether a drug is working or not. And there's also some evidence suggesting, as Dr. Kantoff showed, maybe improvement in time to symptomatic disease. So I think our definition of benefit may be slightly different compared to chemotherapy drugs that can shrink the disease early on. However, I would say that the risk is much, much less than chemotherapy drugs. Just a case in point, we've treated over 600, we've given over 800 doses of vaccine to patients with our ProstVac vaccine at the National Cancer Institute in 6 different clinical trials. We just recently looked at that data, and the adverse event level is pretty interesting. You see about 25% of the patients having a grade 2 injection site reaction, no grade 3. You see about 5% systemic side effects that are grade 2 and about 1% grade 3. Very much different side effect profile than you'd see with chemotherapy, where frequently 85% of the patients will have grade 3 or 4 side effects. So I think overall we're seeing that the risk-benefit profile is completely different to what we see with other tools in our armamentarium.
So I completely agree with that, James. We've treated several hundred patients now with Cipraloy-SLT or with ProstVac over the years, and the toxicity profile is very different than anything we've seen with any of the other agents that we use and hurt people with in medical oncology, including chemotherapy, hormonal therapies, et cetera. I think an issue that really needs to be brought out is this gap between reality and patients' and physicians' perception of what immunotherapy of this sort does. Physicians and patients are used to seeing immediate things happen. Cancers go away. In the case of prostate cancer, PSAs go down, and they're disappointed somehow that that doesn't happen, although it seems clear to me that these are prolonging survival. Now, we've seen this before in oncology. We've seen situations where the benefits are long-term and you see no immediate benefit. In fact, sometimes you see an immediate decrement in quality of life. For instance, adjuvant chemotherapy for a variety of diseases, there's no perceptible benefit immediately, but there's a benefit down the road, sort of an investment for the future. Also, some of the bone protective agents that we use, like zoledronic acid or denosumab, which don't actually have a survival advantage but have a protection against bone-related problems, nothing happens to the patient other than a little bit of toxicity immediately in exchange for benefit later. So we've seen that sort of trade-off before, but it seems to be a new issue again, but it really is an old paradigm of very little is happening now in exchange for the benefits later. And I think we need to do a better job at explaining that to patients because there is so much hype around immunotherapy and a great expectation that the cancer will disappear immediately. And it disappoints physicians, it disappoints patients. So patient education, physician education is going to be very important.
Well, what could I say without duplicating? Just that we are talking about immunotherapies, and you reminded us, Axel, that there are two different immunotherapies, but they are extremely divergent somewhere. There is one that is less specific. Let's say this is non-specific immunomodulation with some toxicity. We could refer back to some decays with interleukin-2, low number of responder patients, but very dramatic responses and long responses. And probably in the future, we will see less side effect because of better targeting or better selection of the targets. On the other hand, you have another immunotherapy, which I would refer to as an active and specific immunotherapy, which emerged from prophylactic vaccines or the active immunology background. And when we talk with doctors or patients about it, they immediately find this approach extremely appealing or sexy, simply because so far there is little or no side effects. If I refer to the MAGE-3 trial, 2,000 patients, repeated dosing, there is no safety signal so far. But one might argue that efficacy is not proven at all. And if you don't have side effects, it means that you don't have activity. We'll see. But so far, it's all for the patients about being able to fight cancer on their own, being able to restore, to re-educate the immune system, and to be able to fight themselves cancer. This is absolutely different from chemotherapy or small molecules. One advantage as well of active immunization is the long-term memory. And if we refer again to side effects, the platform, if I refer to specific immunotherapies, Provenge could be included, but not fully, but others like Stimuvax or other quote-quote vaccines are pretty similar in terms of technology to prophylactic vaccines. And so far, billions of children have received prophylactic vaccines with no or little long-term side effects. So there is no real fear there about inducing after several years autoimmune disorders. Perception about immunotherapy comes also from the other part of the world, not only the patient, the doctors, but also the regulators and somewhere the payers as well. Regulators see it as in a very cautious way, and Philippe, you nicely described all the issues of Dendreon with CIC cell. It's not by chance. It's that to get convinced for a new class of drug, they are probably more demanding than for followers. And hopefully in the next years, it will be probably easier simply because the regulators also learn from all our trials at university or in the pharmaceutical world. The payers do see immunotherapy. They do not see the innovation. As far as I'm concerned, I would think that active immunization or applying a kind of vaccine is extremely sexy. The payers are extremely realistic, I would say, and they see the value and the clinical activity, and they're right, very likely. So they don't dream at all like us. But ultimately, at the end, for the perception, and I refer to interleukin, pegylated interferon with a trial that is globally negative, but in a subset of microscopic disease and even in a subset of ulcerated primary with microscopic disease, there is a trend to see a very few patients benefiting. And the future is going to really depend on how well we are able to define those patients that do benefit from one treatment. And hopefully, for 100% of the patients, we will have several treatments that will address all the pathologies and all the patients. But today we discussed about biomarkers, and this is the main topic. Probably all the uncertainty that is embedded into divergence of the curves or delayed effect or PFS not being correlated with OS. And surprisingly, this is OS that prevails. And usually you see the converse with chemotherapy. All hopefully might be resolved with a better selection and a better understanding of what we do.
So great. So I think that was the first round of perspectives. I'd like to get a bit closer to a few issues. The first one is actually around the differentiation of immunotherapies versus existing agents in the space. So we heard a little bit of that from Vincent just now, but I'd like to turn this more towards our clinicians. How do you feel that the treater community, meaning other physicians that are just getting introduced to new modalities of immunotherapy, so they were not involved in their development, and patients, how do they react towards these new modalities in terms of the true clinical effects? Benefit, meaning efficacy, and risk, meaning safety issues. We touched on the safety mostly, but how do patients and physicians perceive this relative to other treatments?
Same order? Did you put me here intentionally to sort of make me the...
No, there is... Okay, so...
I think this is a really important topic, actually, and I think, let me just, I'm impressed with the overall survival versus disease-free survival observation, and, which seems to be fairly consistent where we're seeing. You have to understand where we're coming from, that the concept of cancer as a disease of cell growth, that the tumor mass is the critical thing, and also the ease of assessing tumor shrinkage from a regulatory point of view, response rate correlating with clinical benefit and so on, has forced us into a paradigm of evaluation of agents based on the concept of tumor shrinkage, duration of tumor shrinkage, which is response duration, and the assumption that there would be an improvement in survival therefrom. I think what we're seeing in modern biology of cancer is a shifting away from mitosis being the primary problem to cell mobility being the primary problem. If you have millions of cancer cells circulating in your body and they don't form tumors, they're never going to bother you. It's not the cancer cell that kills you, it's the tumor mass. And the tumor mass is obviously a complicated thing, involving white cells, involving marrow-derived endothelial cells that differentiate into blood vessels and the co-opting of stroma in the tissues and so on. There's a very complex architecture of tumors, and that's really what kills you, is the destruction of the organ and the recapitulation of this in other sites and other organs, which we call metastasis. It seems to me very logical that if you have an effective therapy that prevents new sites of disease, obviously interferes with the metastatic process, but doesn't do very much for established disease, then you're going to have not very much of an effect on, on progression-free survival initially, because those are sites of established disease. But you could have an overall survival benefit because you're affecting the development of new sites of metastasis, and those could be the problematic things. It's a cliche, I've said it so many times, but you never die of your first metastasis, you die of your last metastasis. And that preventing new sites of involvement could have a profound effect on overall survival, and you're not going to pick it up on the usual methods of assessment in clinical trials based on shrinkage of tumors with anti-mitotic agents. What I find disturbing, therefore, about some of the trials that we're designing is that we're using the same old RECIST criteria for assessment of benefit without awareness of the emerging new biology of cancer in this regard. So, you say that, the patient fails, that they fail because of sites of prior involvement, that they fail because of organ-specific sites of primary involvement, that they fail because of new sites of involvement. These are critically important questions which have to be built into our clinical trials and often are not. And especially when we have company-sponsored trials where they know they're going to have FDA evaluation at some point or another, and the criteria are really set in stone because it's based on what I think is an archaic model of cancer. So what we're really talking about here is the more modern design of clinical trials and awareness of new cancer biology when we're talking about these new agents. And this is many sessions. Matter of fact, in the same hotel fairly recently, there were meetings that were really concerned with innovative new clinical trial designs, and I think those discussions really are very relevant, to what we're talking about at this point. We have a lot of good therapies for local disease. Preventing metastasis, I think, is really what we should be zeroing on, and immunotherapy is perfectly suited for this. And we may actually already be seeing it, and why we're seeing OS and not PFS, benefits. So it might already be there, and that's something we all ought to think about very, very carefully.
Two things. Number one, we are facing how to place these drugs in a specific order. So there are so many new drugs that are now being incorporated into the armamentarium, not only prostate cancer, breast cancer, lung cancer, but I think Larry is absolutely correct by talking about the biology of the cancer, how we use these drugs, how we strategize their placement into the clinical states as the cancer goes from being clinically localized into metastatic, will really be very important into trying to figure out how to avoid resistance. So, for example, we deal a lot with the community oncologists, we're academic oncologists, and very often the algorithm of treatment is very different than what we might be doing in academia. So in the community, they have a very strict algorithm. If you fail A, you go to B, and then you go to C, and then there's chemo, and the first chemo doesn't work, you go to the second and the third. And very distantly on, we're worrying about clinical trials. As opposed to many of the academicians, we like to get people on clinical trials very, very early on. That being said, the concern that is often reflected, not necessarily by the physicians, but by the patients themselves, is the immediacy of treatment results. So while we have all these great new drugs, for example, in prostate cancer, drugs that affect the androgen receptor, the new immune ones, and new chemotherapy, what often competes with a desire of a patient to go on a particular treatment, even if a person is encouraged by their physician, is how fast will they get that anti-tumor effect? How fast will the pain go away? And frankly, they all look at that darn marker called PSA, prostate-specific antigen, otherwise known as promotes stress and anxiety for the doctor. But the reality is that where we get into trouble and where we would lose interest and momentum for some of these other drugs that are out there but may take longer to work is the fact that patients want to be able to take their vacations, they want to go away, they don't want to be hooked up to an IV, and they don't want to know that there's a possibility that their immune therapy is going to take 3, 6, 9 months to have an anti-tumor display on the scans. So while all of us encourage patients to definitely try an immune approach, we are facing issues with the patient. Patients want to see that PSA drop yesterday, and there are other competing drugs that can do so. They want to be able to have a good quality of life. And even if immunotherapy will give them that good quality of life, they still are very reticent to be sitting there wondering when it's going to happen. And then you have the last problem, which is where do you position immunotherapy in our armamentarium? We're very blessed to have all these wonderful new drugs in a lot of different diseases, but the problem is that patients themselves often want to delay going on to those drugs. And so we want to prevent resistance, but patients sometimes are very non-compliant with what we want to do, even though we like to think that we have a very good relationship and a collegial relationship with our patients. So I think it just brings a lot of food for thought here in terms of what we're facing with the real world.
I think that the unpredictability of response, and especially the unpredictability of the time to response, is a real challenge. And it's causing us, for all the reasons that Larry and Susan just alluded to have to, from the very beginning, talk to patients about realigning their expectations about when they might get better. And that's a very difficult thing to do. And so we start to do that from the initial conversation about immunotherapy, that the toxicities may be different and your expectations of when you might get better are different. And what helps a lot is to put a number on it, that X number of people may get worse before they get better. And I think having some of the mystery removed does help people to accept the fact that there is uncertainty. We all know what the answer to this is. The answer is ultimately to find biomarkers that allow you to identify these people up front. And as you heard earlier today, we're all heading in that direction. We're just not there yet. But I think for the meantime, all we can do is educate our patients and their families, and ourselves for that matter, because in many cases it's physicians who don't have experience with this type of biologic therapy that set up the expectations that, well, maybe I'll do a CAT scan after just 6 weeks, and if you're not better then we'll move on to something else. So we all know that no matter what kind of immunotherapy you're talking about, that's probably the wrong thing to do. And so I think it's not just educating our patients and their families, but it's actually educating ourselves a little bit better too.
So I think for me, when I see a patient that comes in, I think I have a very practical consideration. What is their disease doing? What is their pace of disease? Are they having symptoms? If they're symptomatic, if they have rapid pace of disease, I am not likely to put them on an immunotherapy. I'm likely to treat them with something different, unless I'm giving them a combination of immunotherapy plus something else, and we'll talk about that later. If they have a more indolent disease, they're non-symptomatic or minimally symptomatic, then I may offer them an immunotherapy and discuss with them the caveats that we already heard mentioned.
Completely agree. I mean, it's patient selection, it's patient education, physician education, and most importantly, biomarkers that we can use to follow patients. And there's a big distance between biomarkers and overall survival, and everything in between, response and progression, as Larry pointed out, it's really a mess. It's an old paradigm that needs to be, we need to move on from.
I have nothing really to add except that some patients do come back to their investigators by telling, how do you know that this immunotherapy is going to work with me? Simply because for allergy, for diabetes, for other diseases, we are not all equal. So how do you know that my immune system is going to react the way you believe it's going to react? So there is an uncertainty there. That hopefully will be resolved with the new trials going on.
So maybe I'll follow up on Jim. The possibility of, now I think we're seeing more and more combining immunotherapies with either radiation or chemotherapy. So what do you see are opportunities and challenges there that the whole benefit-risk ratio may change?
So I think there's a couple of things here that when one looks at potential for combination, first of all, there are a wide variety of different potential combinations. You have different adjuvants, different cytokines, different immune checkpoint inhibitors and other biologics that may help boost further the immune response. In addition, you have other therapies that could alter the way the tumor looks to the immune system or alter the way the tumor dies in an immunologically relevant manner. So there's a wide variety of different agents potentially that one could use. One interesting thing I'd like to point out is that while To date, we haven't seen an improvement in time to progression with most of the randomized studies in immunotherapy. One might argue that if what we're seeing is an eventual delaying in a delayed immune response that translates eventually into a clinical response, if one could combine it with a standard of care agent, one might be able to see an improved time to progression as the combination of those two agents works together compared with a standard of care agent alone. In fact, we have 3 ongoing clinical endpoint studies, and stay tuned. Hopefully we'll have data from those shortly.
Anyone else about combinations, benefits, risks, changes?
I mean, I live in a melanoma-centered universe most of the time, and obviously there were 2 medicines approved for melanoma last year. They complement each other. One's a targeted therapy, a BRAF kinase inhibitor, and the other is CTLA-4 blocking antibody. And the natural next step is to combine those, but I think that we need to remember that these need to be done as part of clinical trials, not standard care, because we don't know if sequencing is important. We don't know about the potential for overlapping toxicity. So this is, I think, another important thing to stress, especially with some of our colleagues in the community, that just because you have 2 approved medicines for the same disease, that it might not be straightforward to combine them. You know, not as easy as taking CHO and P and making CHOP.
Actually, CHOP wasn't all that easy to make, so obviously. But the, the reflex, A works and B works, so let's do the combination and let's get them, in, simultaneously. I think something else that it's a big issue, and Axel and I were talking about this earlier, is that dose and schedule combinations is critically important. We know this for every agent that we use in every disease that we treat, even outside of oncology. And so, knowing the scheduling, knowing the timing, knowing the optimal dose in combination versus the optimal dose of single agent is not a trivial issue, and I think it's got to be a very, very important part of what we do. And again, I'm just putting in this plug, is that, we've chosen our chemotherapy drugs to kill cells by apoptosis. Apoptosis is what you don't want to do if you want immune stimulation, because you chop up the macromolecules and you're not presenting the native situation to the immune system that it's going to react to. And so one of the things that we're talking about is, can we actually develop therapies that can kill cells by a mechanism other than apoptosis, so that you can get necrosis, for example, which is a much better way of releasing potential antigens. And we've screened out those drugs in the past, because that isn't what we want to do. We want to minimize toxicity of our therapy, so we want the tumor to shrink in an orderly fashion without making the patient ill. And if we want optimal immune stimulation, we may want to have a different approach. So I think the emergence of immunotherapy is a really viable approach to cancer, which is just really very recent and why we're all here, is going to have to make us rethink our cytotoxic therapy as well, and also pay attention to the dose scheduling of those various modalities.
So.
Just one little thing, and that is, I think what Larry says and what James has said is actually underscores the need for some of these neoadjuvant investigations. We're actually administering a combination of different biologics with a chemotherapy or another agent, a checkpoint inhibitor, and you have a fast readout of looking at that tissue after the patient goes to surgery. So you have your own little, if you will, Boyden chamber for cancer. And this probably is the reason why a lot of us are turning toward that kind of approach in order to study these combinations earlier.
Yeah, I would like to just add one more thing here to get to some of the complexity of combination therapies. I think Larry's point is very clear that schedule is important. We haven't paid that much attention to schedule in past investigations with non-immunotherapies. And now actually the universe has gotten a lot larger by having new treatments that are not treating the tumor, but treating the immune system. Then the immune system does the trick on the tumor. Now that basically puts us in the situation that we not only have to look at markers of the tumor or the tumor cells, but also biomarkers and parameters of the host. The immune system is part of the host. So, and we discussed this in some sessions yesterday, that there is a risk that some treatments that are not immunotherapies, like targeted agents, may affect pathways in the tumor, but they may also affect pathways in the immune system and may create a conflict in a way, a different kind of conflict than we had before. So in terms of investigating this, in terms of identifying the right combinations and then adjusting the benefit risk will be a challenge. So are there any comments on that, let's say, a deeper part of the question?
So I think that is critical. I think that you have to look at the impact of the agent you're going to combine directly on the immune cells that you're trying to generate, as well as on the tumor. And hopefully if it either kills the tumor cell in an immunologically relevant manner or alters the phenotype of the tumor, and it doesn't have a significant deleterious impact on the immune system, then I think that there's a potential for combination. And we have taken that approach in our laboratory when we're studying potential combinations.
So as an outsider, maybe I'll ask the panel and whoever out there wants to comment, is that we've seen trials where chemotherapy was combined with an immunotherapy, GVAX as an example, where it was negative. It may have been even worse than negative, in fact. We've seen hints that combining with chemotherapy might be a good thing. From the experts here, what do you think of that combination in general? Chemotherapy plus immunotherapy.
Again, I think it's all depends on the dose and the schedule and the type of chemotherapy you're using. We have a trial that's ongoing with Docetaxel, the same drug used in VITAL-2 that you were alluding to with GVAX. However, we're using it at 30 milligrams per meter squared weekly for 3 out of 4 weeks, and we're sequencing it with the vaccine. We found actually preclinically that if we sequenced the vaccine, if we gave the vaccine the day after the chemotherapy, we'd actually not have a good immune response with this particular vaccine. But if we gave the vaccine the day before the chemotherapy, we could get additive or perhaps super additive benefit. So I think it does matter how you give the chemotherapy, how you give the vaccine, the sequencing, as Dr. Norton mentioned. But, and it may be a difference between the dosing, the higher dose of the Docetaxel. I think also it matters the patient population. I think the patients in the VITAL-2 study were a lot sicker than many patients. So these were patients with symptomatic prostate cancer who were randomized to receive vaccine plus chemotherapy versus chemotherapy plus steroids. And it was trending in the wrong direction before they stopped the study. Those patients on average had a predicted survival of about 13 months. So these were pretty advanced patients. And if one might see a delayed separation in the curves, early events might indicate that it would be worse off for chemotherapy with vaccine compared with the chemotherapy alone. Later on, further follow-up, there appeared to be a less of a discordance between the two groups. They may have been coming together.
This actually begs the question as to whether or not people with significant tumor burdens are the right people for whom this kind of therapy would be appropriate. That's number one. And number two, the number of people who get extra chemotherapies, if they have 4 chemos or 3 chemos, are we really distorting the immune system by virtue of the fact that they've had so much chemo that we are getting rid of the appropriate populations? I don't know if, Jim, in your experience with some of those preclinical models, was there any suggestion that heavy tumor burden would really delay any sort of robustness in the immune system?
I can't generalize on that, but I can tell you some early studies that we did suggested that anti-CTLA-4 worked better against larger tumors than it did against smaller ones. So I think that you have to think about vaccines fundamentally different than you do about things which can use de novo priming. So if you're going to combine something that's killing tumor cells, I think it's best to do it when you've got a pretty significant tumor burden rather than doing it when you've got minimal residual disease, because you're not going to have the antigen to be priming the cells that the CTLA-4 is going to be enhancing the response with. So I don't think we can generalize across the board. But I think it is clear that, you know, if a drug as James mentioned, if a drug suppresses the immune system, doesn't mean you can't use it. You just got to space it out and, let it wash out. And it may have primed some cells. It may have actually done some good, but you can't put something like CTLA-4 right on top of it. You got to wait till it's gone. But not too long, because you need the tumor debris and the dendritic cells to still be there.
So I believe it looked like there was a question from the audience.
Well, I wanted to provide some questions, but also just to comment. I think you're preaching to the choir here about these issues that, in terms of what needs to be done and so forth. So I'd be interested in your comments. I mean, there's so many questions that are unknown. For example, our group was on the extended access trial for ipilimumab, and we had patients on this trial. They had a response, and then later progressed, and then when we tried to put them back on the program, the insurance company reminded us that in the package insert, you're only supposed to give 4 doses of ipilimumab, and that's the end. So I was interested in Jedd's comment on this thing. But more important than that, I think many of us are transplanters. We're experienced in clinical research. I think this is a new paradigm. It's been pointed out. I do believe we have some excitement ahead of us in terms of these new modalities, but I also believe we have some real issues as far as delivering this care with this new paradigm to the patients, in the field, not just in academic centers, but also in community centers. So just some comments about that. I mean, you have a patient that's benefiting from an agent, but then the dose of ipilimumab in Dallas, Texas is $26,000 a dose. And so when you ask a person's insurance company to cover this, and they've already had 4 or 5 doses on the extended access trial, and they say no. So I think we have some things ahead of us. We're studying the anti-PD-1 through the Cancer Immunotherapy Network. I suspect the same thing's going to happen. As I understand it, the cost of delivering the infusion of vaccine for prostate cancer is enormous. But yet we don't have the biomarkers yet. So I think we have some real issues. There's some excitement, but also there's some burdens ahead of us from not only the standpoint of the science, but also from the regulatory issues and from the practical issue of having our patients be able to take advantage of what so many of us have worked so hard to get here. I'm just interested in comments along those lines. We all know that there's lots of work that needs to be done in terms of the combination of agents, the dose and schedule, how these should be administered. We are very excited about this abscopal effect that Jedd and his coworkers commented on. We're also speculating that many patients who get treated for malignant diseases probably have an immune basis for their response and the duration of their response, their cure, in fact, of their disease. So just some comments along those lines. How is it that we're going to go step by step? And I think this organization can, as Axel has pointed out, I think this organization is committed, is obligated to lead the way as to how we bring these new modalities to our patients. They're all over the world. So just your interest, just your comments on those. Thank you. No questions.
So, Joe, those are all excellent points. And to the point that you raised about reinduction, I think it's a more general point of things that we see in clinical trials that may or may not be able to be replicated in the real world. I think that's something that we all grapple with. But our experience is that we know that in the phase 3 trial that Steve Hody published, that reinduction with ipilimumab had efficacy, that although it was a small number of people, there were people who went back into remission, some of them in more significant remissions than they were in before. Mm-hmm. But it is not part of the package insert. And so we've had the same experience, and we get asked by insurance companies this question, and I send them the paper and I write a paragraph, and actually what has helped is that the NCCN has put reinduction into the guidelines, which is very helpful. And so we have not had too much trouble with denial, but it is a challenge that it's not in the package insert right now.
Yeah, there's instances where you have a patient with melanoma that has had a response and then there's progression, and you may irradiate this area, a painful metastatic lesion. And then, based on the publication that's just come out, I think this has engendered a lot of enthusiasm. Our patients read these things.
Yeah.
And so these are some kinds of things. And then the discussion that Jim was giving about, well, if we give this drug at this dose and this, those are very interesting ideas, but we need to figure out how we're going to bring to pass, how we're going to bring this to reality as how we can pursue these. And I think it's different than chemotherapy agents, much different, because as you all pointed out, you could administer cyclophosphamide, erythromycin, vincristine, prednisone and see a response in a week. But you can't administer, anti-PD-1 or ipilimumab and see a response in a week. So this is an issue. But how are we going to bridge that into getting these things available for our patients? I mean, soon. And that's what I think is the biggest challenge.
Thanks.
We had biomarkers, we'd know. If you could tell a patient, look, you have the immunological phenotype that you're going to respond to this drug, be no question about it. So we have to get that data done, but it's going to take some time. We can't even agree on which biomarkers. That's why we're having this meeting, as to what should be done. So we have a lot of challenges there. Lots of nice things are happening. It's exciting, but I would submit that we have tremendous hurdles ahead of us to get this thing to go like we'd all like, is what I would say.
You speak for the choir.
Yeah.
So, and I can certainly say from the side of the consortium that we are committed to identify the issues and do that in a focused fashion, meaning step by step, not everything at the same time, and aim for developing solutions together with you, meaning with the community. Doing it alone or doing it in an isolated fashion has not been successful in the past. I think our track record is to do this with the community, and we continue to do that. So there are some new topics that emerged from this meeting and from some discussions we had with the community. They involve, for example, how do you do combinations the right way? Scheduling is part of that issue, which, as we heard earlier. In addition, tumor heterogeneity emerged from this meeting actually as a challenge that might be something we can address. And there will be other things that will follow suit. So the one thing I can say, based on the comment we just heard, the consortium will continue to be committed and partner with other organizations to provide solutions for these things. And the one advantage that we have as a community organization is we're not timeline bound, which means we don't have to deliver a business result in a certain period of time, therefore choose our projects accordingly. We can choose projects based on what makes sense. And even if there are long timelines, we would start those projects and move forward towards a solution, even if it takes us some time. While in a commercial setting, some things just never get started because they take too long. So I just want to put that out here. Now I'm diverting back to a different variation of the benefit-risk question, which gets us towards the payer landscape. The payer landscape has changed. The economy is not exactly what we all want it to be. Drug reimbursement is getting more difficult. Interestingly, in oncology, drug prices are going up. So this all is difficult to coordinate. So the question I was posed to the panel, based on the best guesstimate you can make, where do you see this going? Will the expectations from the payer side towards benefit risk that immunotherapies will have to deliver in the future in order to become successful therapies and be available for patients, will that change? So and I think I should direct that to Vincent to begin.
Yes. To me, not only because I'm working for a pharmaceutical company, but I would like to address your point and to build actually on your different points, because it's extremely important. We are discussing about combination. Most of these drugs are going to be prescribed also as monotherapies in other settings. So how do you address the combination in the access to the patient? Are you going to price twice? This is something that we should keep in mind when we want to combine, and usually I'm used to telling people combination without emotion, meaning that you don't do your shopping somewhere. There must be a scientific rationale, but you also need to think about the consequences of combining, either increasing the side effects, and then you will have to pay to manage those side effects, and you need really to bring medicines of value that are adding value to the patient, not to other parts of the world. Getting to your points and yours for the biomarkers, it's obvious for everyone we should develop biomarkers. We should select better the biomarkers. But the reason is why. A, because it's ethical. I believe this is the right way to do. If we develop medicines together, I mean, we need to give those medicines only to the patients that do benefit and not to the patients that do not. Not only to avoid the side effects, but simply because this is the way to develop drugs. Two, by having targeted therapies with the selection of the patient, this is the only way to sustain innovation. Maybe I'm biased by the European financial context and the reaction of the European states, but they don't see innovation. If you come with a new class, with a new product, they benchmark to generics, which I can understand because they see only the amount of money they will have to charge. But on the other hand, this is the best way to kill any advances in future medicine in 10 years from now, 15 years from now. Situation not yet there in the US, but you can feel some flavor of it when you have some payer organizations that are setting up their own phase 3 trials to identify biomarkers to select drugs that pharmaceutical companies had put on the market very broadly. I'm fully in agreement with this initiative. We should select really better to sustain innovation. Biomarkers, it has a sense only if you increase your clinical benefit. And this is only natural because if you narrow down your population to the ones that benefit and you have the same clinical benefit, you have no dilution effect, so you should increase. And this is a benefit for everyone. For the patients, they know that they are going to benefit. For the doctors, they know that what they prescribe is going to be efficacious, and also for the pharmaceutical companies because they deliver medicines of value there. And ultimately, the biomarkers, why do we do it? Because it helps everybody, essentially the governments and the payers, in the context of the financial crisis. We can see this in Europe tremendously. The UK organization was the first one to block a little bit the entry of some drugs that have shown efficacy in phase 3, but that were delayed. There are now some new processes in some countries where after the European approval, you have some kind of national approval, and the national approval systematically benchmark with its own view to what is currently available. This has come in Italy, this is going to come in Spain, this somewhere is introduced already in Germany, which is there. And again, in the US, there's going to be a trend towards that. So we all agree that we should go to selection of the patient and biomarkers. The big issue there, I see, is that you need diagnostics to get there. You need to identify, you need a tool. And diagnostic is a commodity, meaning that diagnostic test is cheap. And I don't see how a diagnostic test would cost a fortune in the future. I don't see the reason why. So we need, all together with the regulators and the payers, to find an incentive that goes beyond the scientific appeal. We all agree here because somewhere our rationale is a scientific rationale. But we need also to convince the industry, the regulators, the payers, this is the right way to do. If we, especially when we have safe treatments, like Sipuleucel, like some cancer vaccines, why should we select? Because we don't have side effects. So we need to get an incentive there. So the intent is extremely good, excellent. Practically speaking, there is probably a lot of work to do externally to this community and together with organizations that are not used to the selection of the patients, so that there is a real incentive there, not only a scientific one, but really an obligation to go that route.
So I think, I'll make it brief. I think that we need biomarkers. We need markers that we can use to select patient populations that are going to be likely to benefit, and we need biomarkers that are going to be able to predict patients who are living longer and those who are not benefiting, so that we can not get patients started on the therapy if they're not likely to benefit, and patients who are not going to benefit that are already on the therapy, stop that and move on.
Well, I think we're close to running out of time, and I would just like to say that I think this discussion has certainly piqued my interest and stimulated me to think of what the CIC and CRI should be focusing on going forward. And I think some of these issues about patient selection and also even going into reimbursement and payer type issues are big issues that do impact getting immunotherapies into the standard of care, which is the ultimate goal of the CIC. So I think I want to just thank everyone for being part of this, and I would ask the audience too, we might send out, I think after the meeting, a survey, and we would love to have suggestions of the types of things that CIC can do that really meets your expectations and your needs, so that we want to stay relevant and we want to have an impact in the field. So again, we look to all of you to help guide us in terms of what are the next things and initiatives we take on. So thank you very much.